Okay, so I'm going to try again to start. So I was going to start a little bit with there was a typo in an equation from last time, which is good because now I can sort of cover it again. So last time I was discussing the idea of just how we think about structure in populations, age-structured populations, and sort of building up the general framework for that. Today I'll briefly talk about extending it to stage-structured populations, and then I'm going to present just some very straightforward results for evolution in both age and stage-structured populations. So this is the equation that I was having trouble with. So this is the equation for the sensitivity of the leading eigenvalue, or the growth rate, to a particular entry of the transition matrix. There's the problem from last time. There should have been a J there. So, but it gives me a chance to just sort of introduce this idea again. So again, the transition matrix, let's write out a simple case with like 4 stages or ages. So write like this. The general case, and we've been talking about age-structured populations where, for that specific case, it's a Leslie matrix. So the corresponding Leslie matrix that we were talking about last time. So this specific case only has 2 different kinds of non-zero entries. The fecundities, the reproduction in the first row, and the survivorship probabilities that are in the subdiagonal. So when we ask about sensitivity for the age-structured case, there's only 2 kinds of sensitivities. So if we compare these 2 matrices, we can see that for the Leslie matrix, MI gives A1I, right? And the PIs are AI plus 1I, like that, the way we've numbered them. And so the 2 types of sensitivities are the sensitivity to the reproductive values, the fecundities, and that's the equation that I gave last time, so that was correct. This is just J was missing. And then the other kind of sensitivity is the sensitivity to the probability, the survival probabilities, the age-specific survival probabilities. And again, these equations, they were correct. They were what we had last time. All right, so this is a really specific case. It's nice because we just have 2 kinds of things. And when people think about evolution in age-structured populations, often they're asking a question about if we make changes if there's a mutation that causes a change in some aspect of life history, we might imagine that it changes some of these life history parameters. And then we ask the question, does that mutation increase or does it decrease? What does selection do? So we'll come back to that. This is a very specific case. Today I'm going to just extend it a little bit to the idea of a more general case of a stage-structured population. So the problem is once you leave the simple world of age structure, so the example I gave last time was of those sticklebacks. Arrows should all go there. So the sticklebacks had a very simple life history, relatively. There were only 3 ages, and the only thing that could happen is they could age into the next age class, or they could reproduce and produce stage 1 individuals. And so that's what generates the simplicity of the Leslie matrix. But once you leave that behind and say, well, I'm willing to consider other types of life histories where instead of being age classes, my different stages could be something else. They could be size or some other developmental class. Then it gets sort of arbitrarily complex. So I'm going to give you an example. This is based on a life history of a coral. And again, I'm going to start with drawing the life cycle graph. I need more room. Again, I'm just going to have 4 stages, but now the stages aren't age classes. They're something like size or developmental class. I'll use something similar to this before. I'm going to call stage 1 sexually produced larvae. So, and I'm going to say that the other stages can reproduce sexually. And we can get movement into the different stages. So these might be size classes of a small number of polyps, larger size, and a third size. And then there's some probability, perhaps, of staying in the same stage. But finally, if I'm thinking of something like corals or other colonial cnidarians or other marine invertebrates or many plants as well, there might be other kinds of reproduction. So I might want to include something else. And in this case, I'm going to include fragmentation by budding to produce the smallest size. That's not too much more complicated, but now I've left the world of the Leslie matrix behind. So in this case, we have what's sometimes called, it's sometimes called the Lefkovitch matrix. And for this particular example, maybe I'll draw it like this. I've reversed my indices. That one's right. Okay. What I've done here is I've basically used the different transition probabilities and fecundities from this life cycle graph and filled in the transition matrix. We can write out what the recursion equations look like. So in order to be in stage 1 at time t+1, the only way to get into stage 1, I've made it so you have to leave stage 1. You can't stay as a newborn. So you can just include reproduction from the other 3 stage classes. Stage 2 has multiple ways to get into it. So it's actually quite different. Sorry? Yes? Because I've decided that I'm defining this stage to be sexually produced larvae, and you can't stay as a larvae for more than one of these time units. And so they must either survive and make it to the first size, which is a single little polyp, or they die. They have no choice. So so this is you'll see later that I'm going to do this for sexually produced offspring, because I'm going to separate sexually produced offspring from everything else. So that the only way you can be in that stage is if you've just been treated through sexual reproduction. But that's a good question. Thank you. Thank you. Yes. I feel like this is the equivalent of deriving something, writing the matrix, where I'm like, I should have written them out when you guys weren't in here, then it would have been fine. All right, so for the second stage, you can grow into it from stage 1 you can stay in the same stage from one time unit to the next because you didn't grow, you just remained roughly the same size. Or in this case, because I've defined it this way, the smallest piece could be made via clonal fission from the other two sizes. And then for stages 3 and 4, they're much simpler. For each of them, you either come from the previous size, or you remain from the same size. So, and there's nothing magical about this. This is based on an actual population from the literature, but you can imagine that there are many different kinds of life histories that you could summarize in a life history graph and that you could write a transition matrix for. And so the question is, even though obviously we've lost some of the very attractive sparseness of this matrix, are there still ways that we can use some of the same kind of modeling to say something about evolution in these kinds of populations? And if the answer were no, this would be a very short lecture. Luckily, the answer is yes. So just in general, we talked before about the characteristic equation, right? So if you have any matrix, when you solve this equation, you can find various eigenvalues. And for the case of the Leslie matrix, the solution gives you that special equation, the Euler-Lotka equation that we used last time. There's not a nice general case for an arbitrary transition matrix, but you can find the general form of the characteristic equation. It's written different ways depending on sort of your preference, but the number of individuals in the matrix, I'm sorry, in the vector T, so this is for our specific case. Given by the sum of the roots of that polynomial and then a set of coefficients. These coefficients are determined by the initial conditions. And the matrix, but they're functions of n is 0. And this version of this equation is the version in Caswell's book, I'm basing it on. So we can find, even for a slightly more complex life history, what the roots are, and so it gives us the eigenvalues. And once again, the leading non-unit eigenvalue tells us how fast the population is growing once it reaches, now instead of stable age distribution, the stable stage distribution. So it's the point at which the population settles in a constant fraction of the population being in stage 1, stage 2, stage 3, and stage 4. So since that's true, it sort of leads us to believe that if we can develop a straightforward model for our nice, simple case of how, if we have an allele or a new mutation that changes some aspect of the life history, how that might behave, then we'd be able to do it for this general case too. And so that's what I'm going to do today. I'm going to first just present some results. I'm not going to derive all of it. For the age-structured case, and I'm basing it on Charlesworth's work, and then I'm going to show you the results for a model of the stage-structured case that's my own work. So that's the idea. All right, so I'm going to try. I feel like I use too much of the chalkboard for each pass, and that's why you guys are doing a much better job than I'm doing. I have to, I don't know. I kind of want to leave everything up. I'm going to try the, you can't read. Oh, Caswell, 2001. Sorry, I need to write those bigger, obviously. 2001. So I'm going to try this. See if it will work. I use very little water, maybe it'll dry quickly. All right, so let's think about selection in an age-structured population. And for my notation here, I'm actually following Charlesworth's book, because he's used different notation in different papers, so I had to pick something. So let's go with that. So now we're going to do something a little different. We've had, for an age-structured population, a Leslie matrix, and this has been agnostic, or we have not cared about the genotype or anything. We're just like, this is what it is. But now what if we had a belief that there's a single locus for now, or for all of today, that has an effect on the entries of this matrix? And we might define a new matrix, which is the transition matrix, the Leslie matrix, for an individual with genotype AIAJ at the locus that we're interested in. So it's the genotype-specific transition matrix. In that case, now we might want to track the number of individuals, not just of a certain stage, but also a certain genotype. So we're going to say, we're going to follow let me do this. It's the number of AJ individuals of stage X at time t. And the problem now is that if we're going to project the number of individuals into the future, we have to take account not only of transitions, such as moving from here to here, or staying in the same stage, or going back, where your genotype doesn't change, and so you're still using the same matrix, and sex. Because once you introduce sex, you can change, now you can introduce new genotypes. So we need to take that into account, and the nice thing is, for this case, well, for the age-structured case, and later for stage structure, we'll focus on newborns, because they're the individuals that have sort of a new genotype. So without spending too much time in the details, I'm going to give you the results. And then we'll talk about what I've written. I need to define a few things. So I need to be careful because I'm using lowercase p for allele frequency, so I'm going to have to keep track of things. So lowercase p is the frequency of the particular allele at time t, and I'll use uppercase p's. Don't worry about that, just do this. No. Sorry, I shouldn't have, this has got to be t minus. I won't worry about time for right now, and then I'll just say what I mean, and hopefully that'll be helpful. Before, when we talked about the entries of the Leslie matrix, we introduced the idea of Li being P1, P2, P3, Pi minus 1. So that's the cumulative probability that you make it to age I, right? So in order to survive to age I, you have to survive from age 1 to 2, from 2 to 3, from 3 to 4, et cetera. So we're going to do the same thing here, except we're going to assume that it depends on genotype. So Lij is just the probability that you survive from being a newborn to being of age X. And you have to survive stage 1, and then 2, and then 3, et cetera. And that's what that is, and those are allele frequencies. And then finally, this function B, all right, so let's think about what I've written here. So what we're going to say is that the number of individuals of a particular genotype in stage X at time t are the number of newborns that were born back when this individual was, so if this individual is, sorry, this is not stage, this is age X, t minus X was when this individual was a newborn. At that time, these were the allele frequencies, and this is the probability that that newborn survived all the way to get to X. So what we're doing is we're saying at some point, individuals are made, and the genotype is determined by the allele frequencies, and then we just have survivorship probabilities. Now, one thing that we're doing here is we're keeping track of the IJ and the JI genotypes separately. That's why we don't have like a 2 multiplying that for the heterozygotes, because we're keeping track of the 2 different heterozygotes, the 2 orderings separately. And if I was being very good about this and following Charlesworth very carefully, I would be indicating which was the male allele frequency and which was the female allele frequency, and keeping track just in case they were different in the 2 sexes. But I'm not. I'm going to be sloppy and quick and just assume things are easy, just to make it easy. And also the notation becomes incredibly cumbersome. So we're just going to assume allele frequencies are the same in males and females, and we don't have to worry about it. Okay, so this gives the number of newborns happening at some time, and it's basically we're summing over all possible alleles, so all possible genotypes, all possible stages, and then Kij is this function that gives basically the reproductive value, right? So you have to have individuals that survive to stage X, and then how many babies they make at stage X. That's the total sort of reproductive value of an IJ genotype. All right, so, and that's pretty much the basis of this. There's a lot of possible variants of this model having to do with how you are taking the separate sexes and how you're tracking different things, but this is sort of the easiest case, the simplest case. So if this is true, we can ask some questions about what happens first at equilibrium. There's 2 different kinds of equilibrium to think about. There's sort of there's steady state, not equilibrium, there's the steady state with regard to age structure. And we also might ask, what does it look like at genetic equilibrium? So if we assume we've reached genetic equilibrium, so that the allele frequencies are at their equilibrium values, Charlesworth writes this quantity, a couple different ways. where now this B function gives us the same equation as before, but at the equilibrium allele frequencies. So if we assume that we've also reached a steady age distribution, then the population is growing at a rate that's determined by the leading eigenvalue. And that means any stage is changing at that rate, including the newborns, the stage 1 individuals. So for example, for x, so if we think of this ratio here, for x equals 1, this is the ratio of the number of newborns in time t to the number of newborns 1 time unit ago. And that's just going to be this quantity, which tells us that for just 1 time step, the number of newborns is going to be the number of newborns in the previous generation multiplied by that leading eigenvalue. So it's telling us how it's changing each time step. Could be going up, could be going down, depending on if the population is going up or down. So using this equation then, and this fact that the age-structured population is starting to change at rate lambda, we can define an equilibrium fitness measure. Now it depends on the population being at this point when you've reached the stable age distribution. So we'll consider that it's sometime in the future where that's true. So if we imagine that t is now greater than some t prime, which is the point at which you've reached a stable age distribution, we can define a genotype-specific fitness, equilibrium fitness measure, I put it in this way because this is the notation that's generally used, where they use the log of the eigenvalue. All right, so the question is, how does allele frequency change? So I'm going to use a weak selection. This is under weak selection. And I need to define what I mean by weak selection. Now, I've erased my transition matrix, but I'm going to define weak selection with regard to how much my transition matrix changes if I have a different genotype. So you can pick any genotype as an arbitrary standard and then say, for that standard genotype, the reproductive function, K, is again the product of the probability of surviving to age X and then the fertility or the fecundity of age X. And if that's true, under weak selection, then what we're going to say is the reproductive function for any genotype, any other genotype, is just the reproductive function for whatever one we've picked as our standard, plus some small value epsilon. Sort of formally, the absolute value of this is less than some small amount for all I, for all j, for all x. And it's this value here is a positive number that measures the strength of selection, and it's just weak as long as order, entries of order epsilon squared can be ignored. respect to values of order epsilon. So for everything I'm going to do for the rest of today, I'm going to be assuming this weak selection. So we have to do that in order to make the approximations that let us look at how allele frequencies change. So we're going to say we have weak selection, and then ask the question, how does the allele frequency change in this age-structured population? We're going to sort of jump to the answer. And you're going to see that you've actually seen this, well, a version of this equation by everybody. All the speakers have shown this equation, I think, at this point. Well, not this version, but you've shown a version of it. And I'm going to write it sort of two different ways. That lets me introduce another idea that I want to talk about very briefly. All right, so I've written it this way with that t function in there because I want to remind us that we've actually seen this before. Now this is the version of t for an age-structured case, but earlier in lecture 1, I wrote the case where we have an unstructured population. I wrote this. I wrote a version of this. I actually wrote it in terms of Lx and Mx. I wrote it like this. But those two things are equivalent. And I said this was a measure of generation time because it gave the average age of parents when the population was at a stable age distribution. Or, well, it actually just gave the average age of parents because that was the unstructured case. That's what this gives here. This gives the average age of the parents of newborns when they are at stable state distribution. Oh, sorry, there's a summation missing. So we have to sum over all the genotypes. And that lets us see that, quite simply, all we're doing here is we're replacing this reproductive value that we had before in our unstructured case that only depended on the generation, on time here, with a case here that it's genotype specific and then just summing over the frequencies of those genotypes. So all we're doing is taking an average over all genotypes of the generation time. So this is one sort of formulation of this. If we go ahead and say, well, I'm going to approximate this marginal fitness. Well, actually, I'm going to define the fitness here with regard to this rate of increase. And I'm going to take a Taylor series expansion. I can rewrite this the following way. And this is the equation you've seen now a bunch of different versions. And we define an intrinsic rate of increase for a population of AIAJ individuals at the steady age distribution. And then let R bar be the average of that over the genotypes in the population. So we've seen at least most of this before, right? So basically what we're doing is we're saying an allele AI will increase if the marginal intrinsic rate of increase for that allele is greater than the average, the global average, averaged over the steady state, averaged over the stages, the ages, and averaged over the genotypes. So we saw something very similar to this earlier, and I just want to write over here the classic diploid unstructured case for two alleles. This is what you would see in any population genetics textbook. So assuming random mating, so that you could write the mean fitness this way. So this is sort of the classic result for an unstructured population. You've seen versions of it. So here we have the example for the age-structured population, and it basically says that under weak selection, the genotype-specific intrinsic rates of increase, these, play the same role as the genotype-specific relative fitnesses, the WIJs. And these, of course, are functions of the lambdas, right? So we're saying that under weak selection, you can actually get the same sort of result that you got for an unstructured population. You make some assumptions about how you reached the steady age distribution. All right, so that's straightforward, and it didn't depend too much on the simplicity of the model, although it did a little bit. So now the question is, so that's an old result. The question is, can we do something similar for the slightly more complicated case where we have a population of organisms that isn't an age structure, that maybe stay in the same stages, that have clonal reproduction and sexual reproduction, and how can we do that? And we can do it very similarly. I'm not going to show all the sort of gory details, and I'm actually going to change notation a little bit. Yeah, it's not so nice. Because all of a sudden the IJs are going to go to a superscript because I need space for other things. But hopefully you'll be like, oh yeah, that's not so weird. So if I had a whole nother chalkboard and I could just push this up, that would be perfect, because then I could leave that there and then write underneath it. But I'm not going to do that. But how about if I write the next piece right here? So the first two bits could be together for a second. Let's do the same sort of thing for a stage-structured population. So now we're going to say the same thing. We're going to say that we have a genotype-specific transition matrix. The only thing is I'm going to lift the superscripts. And those brackets, when I use them, don't mean a global average. I was using them differently. Yes? Yes. Yes. Yes, it's general. There's nothing in this that says how selection acts. It's just that it's weak, and that the action of the locus is to change the transition matrix so it changes the growth, the intrinsic rate of increase, the leading eigenvalue. That's it. So it could make it better, it could make it worse. I mean, it could do anything. So can you see things like heterozygote advantage? Yeah. I mean, yeah, it's as long as you're just dealing with the simple case of a locus with 2 alleles. Charlesworth shows that you can pull out almost like all those things come out. And once things get, I'm not sure what happens if you do multiple alleles. To think about that, but yeah, it's completely general for as long as you have weak selection. If you leave weak selection, then you have problems because you can't define the fitness the same way. So basically, if you think about it, what you're basically doing is you're saying, instead of saying each genotype has a fitness, you're saying each genotype has a matrix which determines its fitness. That's all you've done. So you've just made fitness a more complex trait. But fitness is a complex trait anyway. You've just divided up the complexity of it in a certain way. And that's what we're doing here as well. We're saying that, whatever the transition matrix is, it's defined by the genotype at this locus. And so, in the same way that we wrote this, we can write something like this. But now time is going to do funny things. Or funnier things, anyway. All right. So now there's new things that have shown up, and that summation got kind of strange. And I feel like there should be... No, that's fine. So here, there's no summation over time, because if an individual is of age X in an age-structured population, you know exactly how far back in time you have to go to find those newborns. You have to go back X time steps. I've erased my life history graph, but one of the features of a life history graph that is not age-structured, where you have those self-loops where you can stay in the same stage, is that individuals of a particular stage don't have an age, they have a distribution of ages. They have a distribution of ages if we define age, the way I did in this model, as the time since they were a newborn. So we have to find some, somewhat arbitrary, but we have to find some place to plant our flag and say this is where time starts. And I'm saying time starts when you're a newborn for a stage-structured population. I'm separating out that stage. And I want to know how long ago it was. Well, that amount of time depends on how many steps of clonal reproduction there's been, how much I've stayed in the same stage. But you can easily find how long it's been. Well, you can make stage 1 an observing state and just figure out, on average, what for each stage the absorption time to going back to stage 1 is. And that's basically what you do. There's a couple of different ways. I did it 2 different ways in 2 different papers. Why, I don't remember anymore. No, I do remember. For one paper, I found it with an assumption that you'd reach steady state, and for one, I did it where you didn't make that assumption. Let's think about that for just a second. This part's straightforward, right? The number of individuals that are newborns, stage 1, at time t is just summing over all the stages, that should probably be outside, all the stages and all the genotypes, everybody that was around in time t minus 1, and this is just the entry of the transition matrix that's producing newborns. This is the equivalent of the MIs in the age-structured case, right? So this is just all the production of newborns by everybody. That's what dumps into this. The question is, what does this mean? So this is the number of newborns that were around tau time units ago, and we're going to sum over all possible tau. Tau is how far back. If I'm in stage X now, how far back in time do I have to go to be a newborn? Well, that's determined by this probability. So let me describe what that is. how do I determine how old I am if I'm stage structured? Well, I'm just going to have to figure out what's the probability of going back 1 step, 2 steps, 3 steps, etc. So one way, which maybe is the easier way, so maybe I'll just go ahead and use that one. So I'm going to define this function. And that's the probability of a stage X individual being tau time units old, which is also the probability of moving from stage 1 to stage X without going through stage 1, right? Because stage 1 is an absorbing state. If I go into stage 1, I've got to stop. In exactly tau time steps. There's a couple ways to do that. I'm trying to decide. I think I like this one better. So I can define another matrix, a slightly different matrix. And this matrix is going to give the probability of being in stage L 1 time unit ago, given you're in stage K now, and we're going to make 1 an absorbing state. So this is going to be a Markov matrix. If the population is at the stable stage distribution, then the entries of this matrix, and this is, I have to say, this is, I feel like I should, this is like one of those things with an advertisement that's a warning to young people not to do something stupid. Never use L as a subscript for any reason, because it always looks like ones in many fonts, and then it's a really terrible thing. I used it in a whole bunch of papers, and I regret it bitterly, but now the habit is there, and it's very hard to change. But don't do what I do. Yes, yes, or do that. One of those two things is what you should do. You have these L's and these J's. It's horrid. So notice that my N's no longer have a time dependence. That's because I'm now at stable stage distribution. And then a couple of things. I have to define 1, 1 here as 1, and 1L for all L not equal to 1 as 0, so that 1 is an absorbing state. So I basically make the Markov matrix version of my transition matrix with 1 as an absorbing state. Once I do that, I can just basically ask for sojourn time. How long before I get absorbed into stage 1 on average, depending on where I start. The probability, given that I'm in stage K right now, that I went back to stage 1 by tau time units ago, is going to be this quantity. It's sort of horrid. This is this matrix raised to the tauth power, and then the K-elf entry of it. So that gives the probability. And then if I want to, but that tells me how, I've done it by time tau. I want to know exactly time tau, right? I want to know how I'm tau years old. I want to be certain, and I've got to just do this. So this is from X to 1. I want to know, I want to know what's the probability of going from X to 1 in exactly tau time units. That's this quantity. So this is. So once I've done that, I've defined basically an age for stage X individuals. So it's just this absorption time. There's another way to do this that doesn't depend on the steady state, which is maybe a little prettier, but I feel like this one's more intuitive. So but it points out a really important difference between stage-structured populations and age-structured populations. And that's the fact that you have indeterminate growth and that leads to a distribution of ages for each stage. So individuals of a particular stage were last a newborn some distribution ago, with some mean and such. Okay. So that then tells us what this equation is saying. It's saying that the number of individuals who are in stage X at time t of a particular genotype, we're going to go back in time over all possible times that individuals who are now in stage X could have first arisen. Look at the number of newborns and just sum over all the genotypes. So we're going back and counting up all the genotypes. So I'm trying to, oh, I did erase some stuff. Oh, well, that's fine. So we can do the same sort of thing as we did for the age-structured case and say, what's the equilibrium fitness? We can define it this way. And here, this lambda bar is the leading non-unit eigenvalue for the population at the equilibrium allele frequencies, the p-hats, the same way that we defined it before with r with a little hat over it. So now we have this contribution of an AI-AJ genotype at the newborn stage to the whole reproductive value of the population. So what we can do then is do the same sort of thing that we did for age-structured case and ask the same question. We have this matrix, which I've erased, and it determines things like how long, it determines the distribution of ages for each stages, it determines the fitness, it determines all the aspects of the population. And we can ask, if we have a single locus that affects that transition matrix, if we introduce a mutation or a new allele that has some small effect on it, what's the probability that it'll increase or rare? What's the change in allele frequency? And I very sadly have erased one of the things I wanted not to erase. If we once again assume weak selection, and I won't write out the details, but it's basically the same assumption as before, that setting some genotype to the standard, that any genotype differs from that by some small amount epsilon. We can then define the change in allele frequency at stage 1. Where now we have this function. It's like the t from before, but I had to give it a different name because it's not quite the same thing. This is a generation time, but it's a very specific type of generation time. It It has the same sort of overall shape as the generation time for the age-structured case, but this tau is overall time. Now, in reality, these probabilities, remember this is a distribution, right? This is the probability that an individual in stage X was last a newborn at time tau. And if you took any reasonable stage structured populations transition matrix and went ahead and calculated those probabilities for 1 through 100, 1,000, something like that, it depends on what those age steps are, you'd find that as tau gets bigger and bigger, these probabilities become vanishingly small. I mean, technically, this is infinite, right? There's some tiny probability that you're just looping around forever, but that's not really true. So if you're actually calculating these sorts of things for a numerical example, what I usually do is just cut it off at some, you can calculate them and look at them on the computer, just cut it off at some reasonable time. But this is what I've defined, and other people have used also, as something like a meiotic generation time. So it's an average time, if you like, the average age or the average amount of time from sort of meiosis to meiosis. So going back to stage 1. And remember at the beginning I said, or actually someone asked me, they said, why is stage 1, you can only get in, there was no self-loop for stage 1. And this is why. I want stage 1 to be this absorbing state, that when I go there, that's sexual reproduction, and I'm counting that as time starts there. So this generation time uses that definition to say time starts with sexual reproduction. So doing a similar sort of approximation as we did for the age-structured case, basically I do the same sort of Taylor expansion, but I won't actually go into. We can rewrite this, and now it's going to look like the same equation we keep seeing all day today. It's sort of nice, I guess. Multiply that. So, similarly to what we did before, we're going to define a set of eigenvalues that are at the steady stage distribution, how the population grows, that has a particular genotype. It was all the same genotype. And then we take the weighted sum of that, lambda bar. And basically, this is what this is saying is the allele will increase if the marginal geometric growth rate is larger than this average. And you can see that that is exactly equivalent to the case for the age-structured population. So indeed, we can, as long as we make the same sort of weak selection assumptions, we can do the same thing. And we're basically saying that, once again, the rate at which the population grows at the steady stage distribution is the equivalent of these genotype-specific fitnesses for the unstructured case. So as long as we're operating in the world of weak selection, this lets us do that. So that's nice. So, okay. But why do we want this, right? Why do we specifically? Well, one of the reasons that people have done this for both the age-structured case and the stage-structured case is to ask questions about life history evolution. So we might ask the question, how do alleles that make changes in different aspects of the life history, are they likely to increase when rare? if we think of sort of trade-offs. So just one, let's think of the simple example of two alleles, A1 and A2. And ask the question, when does A2 increase? Because we might imagine a population fixed for A1, and we introduce a new mutation A2 and ask the question, does it take off? So if we go ahead and we need this is obviously, positive, so we need this to be a number greater than 1. So we can write this, we can rewrite that condition this way. Sorry, 1. Which simplifies to this condition. All right, so what do we need? We need this inequality, we need it to be greater than 0, and we can see that what it depends on is how the rate of increase on those for the transition matrices, it's basically this difference between the heterozygote that carries one copy of the A2 allele and the homozygote that has none. And I think I have made a mistake. It should be 2-2, right? Yeah. And then the case where you have two copies of the introduced allele and just one copy. So, so there's a few different ways then to go about that. One way to do it is to ask the question, to ask a very specific question about how different these two things are likely to be. I'm going to work through a little example. I think it's helpful. Yeah. So we imagine a population fixed for A1, and we introduce, so it has matrix A11, it has geometric rate of increase of lambda 1, et cetera, and we introduce an allele, A2, that affects let's say it affects a single life history parameter for right now. XY. So it's just one of the entries of that matrix. We can approximate the lambdas using Taylor's theorem and write something like this. Again, this is a weak selection case. I'm not going to go through the hairy derivation of this. And now we're like, that's not so bad, because what this says is what we need to know is how much it changes the entries of the transition matrix, and then we need to know the sensitivity of 1, of the leading growth of the geometric rate of increase to changes in that particular entry of the transition matrix. And that's what we started today with, right, is finding those. And so we can rewrite this, obviously. approximately. So we're going to ignore the terms of order epsilon squared. And then we can do the same thing for when we have 2 copies of the mutant. And now it's the difference between those 2 entries and now the sensitivity of the heterozygous matrix to that particular entry. Plus, yeah, things that we're going to ignore. All right. So we can also show that if A2 affects a set S of life history parameters, we can show that the same sort of thing is true. So I can just write this one for here. So this is all of the life history parameters that it affects. The difference in the particular entries between the heterozygote matrix and the homozygote sort of wild type matrix times the sensitivity. And a similar equation for the other case. So now we have these 2 entries, right? So I have time, I have 18 minutes. You have 18 minutes. We're going to do something. Let's see if this works. I'm going to give you a problem. It's on the handout. So I'm going to have you tell me if something's going to increase when rare for a particular case. Okay, so this is a adapted life history. I'll actually draw the picture here. We make things simple and just have 3 stages. It's similar to what we just drew. Stage 1 are newborns. Clonal reproduction here. Here's A. Good if I don't make a mistake now, near the end. Here are the actual entries for this. This is for, this is adapted from this colonial hydrozoan. Okay, that's the transition matrix. I cheated, I used Mathematica, I didn't do this by hand. The leading eigenvalue for this is a growing population. The right eigenvector for this leading left. I want to know if I introduce an allele, A2, it increases survival for stage 2 by amount alpha, and it decreases survival for stage 3 by an amount beta additively. So I'm going to show this. So here we have the genotypes. And here are the 2 survival rates. The survival rate from 3 to 2 and from 2 to 1. And so this is some baseline. And then one copy increases it by amount, this one by an amount alpha, and decreases this one by an amount beta. And in this case, I'm having it be additive. Although you'll see that that's not going to matter in a minute. Okay. So I want to know if this is going to, under what conditions would A2 increase when rare? So we could make things simple. We could make things complicated, we can make things simple. We could say, let's take the extreme case that A2 is very, very rare and A1 is approximately fixed. So we could say A1 is approximately 1 and A2 is approximately 0. And yes, I have erased everything important that I was supposed to not erase. You guys should have stopped me. This is the condition. We want delta P2 to be greater than 0. So we need P1 over the set S of these. Plus P2 times, something. But I'm going to let P2 be approximately 0. So I'm going to ignore this. And I'm going to let this be 1. So this is my condition. I need that to be greater than 0. Okay. Get your paper out. You guys are going to calculate this. Or you're going to figure it out. Let's figure it out together. What's the first thing I need to know? This part's easy, right? I can just figure out, I can figure out the sensitivity, right? For the two stages that are in. There's only 2 stages, luckily. So the XYs, there's only 2 of them. So what I need are 2 sensitivities. And then I just need the difference between the entries of the stage-specific transition matrix for those 2 entries. So does anyone remember how we find the sensitivities? Yeah, maybe from the beginning of lecture? The equation I had a mistake in. So there's 2 sensitivities we need. Oops, no, sorry. It's the 2 stages are 3, 2, and 2, 1. All right. So those 2 sensitivities. So what we need is these numbers over here. Right? The left and right eigenvectors. So I'll leave it to you to calculate these as an exercise, but the answers are, totally did this beforehand. This one is the bottom one. And this one is my top one. Hopefully I didn't mess those up. It's 3, 2, 3, 2, and 2, 1, 2, 1. Okay, so then I just have to say, well, what's the difference over the set? I have 2 differences. I have the difference between this and this, and this and this, right? So it's obviously plus alpha and minus beta. So the condition I need is alpha times this. minus beta times the other number, 28.95, to be greater than 0. Hopefully I haven't. This is the other thing you should never try to do in front of a chalkboard, is do any kind of actual math. That's a ridiculous thing. Don't do that. If I haven't inverted anything or messed anything up, that ratio needs to be 20, more than 20. So the increase in survival is sort of insane. It needs to be 20 times higher, right? For stage 2 to 3, than the decrease. So this is a case of this extremely unlikely, right? And it makes sense if we go back to the transition matrix, which I totally erased. There's very low survivorship from newborns to stage 1. So there's really, it'd be very hard to imagine a situation unless you increased something else dramatically, where decreasing newborn survivorship would be favored in this population. So this is a little bit of an extremely silly example, but I needed to do something quickly, so I did it. So where do people use these sorts of arguments? I've used them before in looking at the evolution of senescence. So senescence is generally defined, well, defined different ways, but it's usually defined as a decrease in either fecundity or survivorship or some other life history attribute with age. Senescence is a little weird to define for these guys, right? Because age becomes a little bit different. But we could also ask the question as, are trade-offs later in the life history stage in general, is selection weakening as you move through the life history, even for things that are stage structured. And in general, for age-structured populations, right, that's the reason that one of the reasons that people discuss about the evolution of senescence is the strength of selection goes down with age. And some mutations that do these kinds of trade-offs, that trade off late survivorship to early survivorship, are usually favored. Did the opposite. What I found that in what other people have found as well is when you look at stage-structured populations, because later stages can actually, you can end up having high fecundities and staying in those stages, you don't necessarily see that kind of trade-off, which has been an argument for why you may not see the same type of senescence evolving in things like trees and other things that have indeterminate growth and have high reproductive value later in life. So I have a few minutes for sort of questions, but that's about as far as I wanted to go. And you can sort of think about other cases where this sort of analysis is something that people do. And so you're basically asking the question, you introduce a mutation that changes aspects of the life history, and under what conditions would that be favored? And this lets you do this. This is general for any kind of stage structure population. Okay, any questions? Well, yeah. I just was being a little bit, I was hedging a little bit because it's clear in an age-structured population what I mean by senescence. I mean that either the L of x's or the M of x's go down as x goes up, right? Here, like, do I mean that for the different stages, so as I get to bigger stages, or do I mean it for the mean age per stage, that distribution? I mean, you could define it either way, and both would be fine. There's nothing wrong with defining it either way. But the main question is, you can think of things early in a life history, and you can think of things that happen later in the life history, even if exactly when they happen has a distribution. And you can ask the question, if I have a mutation that causes a trade-off between early and late in the life history, is it favored or not? And that's the basic question anyway. And I'm certainly not the first person to sort of do this kind of analysis. People have done it in different ways, looking at both. There's a lot of organisms that have indeterminate growth that aren't things like trees and stuff. Fish, a lot of fish have really high fecundities when they get older. They get bigger and bigger and bigger. It's not too surprising that you don't see sort of evidence of senescence in some of those species. Because they seem to have, reproductive functions that increase with time. Other questions? Are you all ready to work out the problem? And yeah, I'm going to work it out and turn it in. Hopefully that was somewhat clear today. A little bit less jet lag today, so it was a little easier. Okay, I think we're fine. I think we've got like 5 minutes, but I'd be happy to answer questions if you guys want to come down, but you can be done.